lipids and nucleic acids. Okay, lipids and nucleic acids, these are examples of haptens. So they alone cannot induce antibody formation. Okay, so kailangan nila ng protein. So example, lipids plus protein, magiging lipoprotein na sila. Okay, so kapag maging protein sila, they will now induce antibody formation. Okay, so lipids plus proteins, lipoproteins, nucleic acids plus proteins, nucleoprotein. Then now they are all immunogen. Okay. So what are the different traits of an immunogen? So pag-usapan natin yan. So what are those different traits of an immunogen? First, we have here your molecular size. Okay, so first we have here your molecular size. So always remember, the greater the molecular weight, the more potent the molecule is. So the higher the molecular weight, the more immunogenic. Okay, so the higher the molecular weight, I repeat, the more immunogenic. Okay, so dito, size does matter. To become a potent immunogen, kindly take note, you must have, okay, so to become a potent immunogen, you must have at least 10,000 Daltons. Okay, so to become a potent immunogen, you must have at least 10,000 Daltons for you to be recognized. Okay, so again, size does matter. So the bigger the molecular weight, the bigger it is to be recognized by the immune system. Another is complexity. Okay, always remember, the more complex, the more immunogenic. Okay, so the more complex, the more immunogenic. Okay, so the best immunogens, we have here your polysaccharides and proteins. So the best immunogens, we have your proteins and polysaccharides. So take note, proteins is these are the most complex substance. Okay, so proteins, these are the most complex substance in the body. So hence, these are the most immunogenic. Okay. Next, less immunogenic, we have here your carbohydrates. So less immunogenic carbohydrates, and not immunogenic, we have nucleic acids and lipids. Okay, so we have here the non-immunogenic, we have here your lipids and your nucleic acids. Bakit sila not immunogenic? Because remember, these are all haptens. Okay? So I repeat, these are not immunogenic because these are actually all haptens. So kailangan po nila ng carrier molecule for them to what? Kailangan pa nila ng carrier molecule for them to become immunogens. Okay? So ito parang sa cellphone. Okay? So complex, mas madali siyang makita ng mga magnanakaw as compared to the simple. Okay? So mas madaling makita ng mga magnanakaw yung complex as compared to the simple one. Okay? Next, foreignness, or the more foreign, the more immunogenic. Okay, so take note, the more foreign, the more immunogenic. Okay, so one of the primary reasons why we do not give we do not perform organ transplantation. Okay, sir, bakit? Kasi kapag other organs, it came from other species, then they are usually immunogen, or they may induce an immune response. Okay. So these are the different types of antigen. So these are the different types of antigen to the host. We have here your autoantigen. These are antigen derived from the same individual. So autoantigens are antigen derived from the same individual. Remember, this is what? Pag sinabi natin autoantigen, these are your self-antigen. And autoantigen should not be immunogenic. Okay? So autoantigen, normally, they are not immunogenic. Bakit? They are not immunogenic because, okay, they are not immunogenic because remember, this is our own antigen. So dapat hindi siya nag-induce ng immune response. And the ability, that is what we call as self-tolerance. Okay? So, the ability of our immune system to accept self-antigen, ang tawag po doon, okay? Ang tawag po doon, self-tolerance. So, the ability of our immune system to accept self-antigen, again, it's referred to as your tolerance. Okay? Alloantigen or homologous antigen are antigen derived from different individual from the same species. Okay? We have your heteroantigen or synogenic antigen. So, these are antigen derived from different species. And remember, of all of these antigen, this will elicit the greatest immune response. Okay? So, of all of these antigen, this will elicit the greatest immune response. Okay? So, I repeat, this will elicit the greatest immune response. Okay? So, itong antigen na to, this will elicit the greatest immune response. Pang-apat, ito, we have here your heterophil antigen. And heterophil antigens are antigens found in unrelated plant or animals. Okay, they cross-react with other antibodies. Okay, so they are found in unrelated plant and animals, and they cross-react with other antibodies. So again, the one that will elicit the greatest immune response, that is your heteroantigen. Next, we have here your graph. Pag sinabi po natin graph, we are talking about transferred part. Okay, so pag sinabi po natin graph, we are talking about a transferred part. So we have here, take note, we have here your autograph. So this is a graph derived from the same individual. Halimbawa, may butas yung pisngi mo, kumuha ka ng balat sa puwet mo. Okay, so that is an autograph. And normally, it is not immunogenic. Next, isograph or we have here your syngeneic graph. This is a graph derived from different class but identical individuals. So this is coming from your identical twin. So take note, this is coming from your identical twin. Allograph, this is found on different individual but same species. Okay? Actually, this is the basis. Okay, so this is actually the basis of transplantation. Okay, so I repeat, this is the basis of transplantation. Okay, so ayan yung allograft. So this is the basis of, because remember, same species, ano yung priority na? Dapat organ should be coming from the same species. Okay? Next. And heterograph. Okay, so these are actually graph derived from different species. So take note, these are actually graph, I repeat, derived from different species. So xenograph is also referred to as your heterograph. Okay, so xenograph, I repeat, this is also referred to as xenograph. Next, so another major topic is we have here your major histocompatibility complex. So we have here your major histocompatibility complex. Kindly take note, this is a set of genes that controls tissue compatibility. So the major histocompatibility complex is actually a set of genes that controls tissue compatibility. Okay, so this is found in chromosome 6 at band 21. And in human, this is referred to as your human leukocyte antigen. So in humans, I repeat, this is referred to as your human leukocyte antigen. Okay? So in humans, also another definition of MHC is that this is the molecular or basis of self from non-self. So take note, this is the molecular basis of self, okay, from non-self. So this is the reason why this is the reason why our immune system can recognize foreign from self antigen. So this is one of the reasons why our immune system can recognize our immune system can recognize self from foreign antigen. Okay? So there are three classes of MHC. So class 1 and class 2. These are actually involved in antigen recognition. Okay, so class 1 and class 2 are actually involved in antigen recognition. Okay, so these are actually involved in antigen recognition and presentation. Whereas class 3, this is actually associated with the secreted protein. Okay, so particularly example na may secreted protein, we have here your the complement protein. Okay, so we have here your complement protein. So example, with an immune function, we have here your complement protein. Okay, so class 1, class 2, associated with antigen recognition presentation. Class 3, ito po yung minor, ito po yung associated sa complement proteins. Okay? So, let us now differentiate class 1 from class 2. First, we have the MHC class 1. Okay? So, pagdating sa class 1, this is expressed in all nucleated cell. So, all cells, actually, basta, generally speaking, basta may nucleus, it has the MHC class 1. Okay? And the highest concentration of the MHC class 1 is found in, okay, the highest concentration is found in the, they are actually found in the lymphocytes and myeloid cells. So the highest concentration, I repeat, these are actually found. Okay, so these are actually found among. Okay, these are actually found among lymphocytes and myeloid cells. Okay. What else? Okay, so low concentrations, these are found in liver, hepatocytes, neural cells, muscle cells, and sperm cells. Okay, so I repeat, this can also be present among low or undetected on liver, hepatocytes, neural cells, and muscle cells and sperm cells. Okay? So genetic locus, we have here your HLA, letter A, letter B, letter C. Okay, o basta letter A, letter B, letter C, that is your MHC class 1. And presented, the antigen is presented to the what? To the T cytotoxic cells. So the antigens, I repeat, these are actually presented to the T cytotoxic cells. Okay, so antigens are present. Remember, pag sabing T cytotoxic cells, we are talking about the CD8 positive cells. Okay? The chain structures, take note, the chain structure, we have the alpha chain and the beta 2 microglobulin. Okay, so the alpha chain and the beta 2 microglobulin. Now, we have the MHC class 2. Pagdating naman sa MHC class 2, this is present in all macrophages. Okay, so this is present in all macrophages, B cells, dendritic cells, and antigen-presenting cells. So, ang tawag dito, take note. Okay, so ang tawag dito is, okay, these are the immunocompetent cells of the immune system. So, these are the immunocompetent cells. Okay, so immunocompetent cells of the immune system. Okay, so ayan, so immunocompetent cells of the immune system. In mayhon, ang tawag nila dito, ay mayhon, kay Stevens, ang tawag nila is the antigen presenting cell. So take note, see the difference. Yung class 1 in all nucleated cell. Okay, pero pagdating sa class 2, okay, piling-pili. Okay, so this is found among what? Immunocompetent cells of the immune system. Okay, so immunocompetent cells of the immune system and also found among antigen presenting. Bottom line here, okay, ang B cells, it has, as what I have mentioned, it has MHC class 2. Okay, so it has MHC class 2, whereas the T cells, wala. Okay? So, kindly take note of that. The B cells contains MHC class 2 pagdating kay T cells, wala. Kaya that is one of the way to differentiate this lymphocyte. Okay? Next, HLA, the antigen is presented to the T helper cells. This is your CD4 positive cell. Okay? So, this is your CD4 positive cell. Remember yung class 1, it is presented to the CD8 positive cell. Ito CD4. Okay? Next, the genetic locus, HLA-DP, DQ-DR. Pagdating kay class 1, it's HLA letter A, letter B, letter C. Okay? So, letter A, letter B, letter C. Ito basta D, that is actually class 2. Okay? Next, we have here your MHC class 3. This is a minor type of MHC. It involves, as what I mentioned, this is associated with complement components. Okay, so complement components such as C2, C4, C4b. Okay, and what is associated to the what is the blood group associated to this is we have the Bennett-Goodspeed. So the blood group associated to this particular MHC, we have the Bennett-Goodspeed. Okay, so now we have here your HLA typing. Okay, so HLA typing. So the process of determining the HLA type of the HLA type of the cell, it is referred to as your HLA typing. Okay, so it could be phenotypic or it could be genotypic. So phenotypic, this is the process of determining the phenotype. Genotypic is the process of determining the genotype. So HLA, there are two types of HLA typing. It could be phenotypic or it could be genotypic. Okay. So first we have here your okay, so it could be what? For HLA class 1 typing, o yung sinabi ko, okay, so for this particular HLA typing, we can use both the B cells and the T cells. Remember, sir, bakit lymphocyte ang sinasabi mo? Always remember, the cell with the highest concentration of this what? The cell with the highest concentration of this HLA, that is your lymphocyte. Okay, hence, we can use the lymphocyte in what? We can use the lymphocyte in performing the HLA typing. Okay, sila may pinakamataas na HLA. Okay, so sila may pinakamataas na HLA. Okay, but for class 1, we can use both B cell and T cell. Okay, so for class 1, we can use both the B cell and the T cell. Okay? But, for class 2 testing, okay, we can actually what? We can actually use, okay, only the B cell. So for class 2 testing, I repeat, we can only use the, what? We can only use the B cell. Okay? Kasi as what I have mentioned, class 2 is present sa B cell but not in the T cell. Okay? So first, let's talk about HLA phenotyping. HLA phenotyping is based on what? HLA phenotyping, kindly take note, this is based on the complement-dependent cytotoxicity. Okay? So this is dependent on the complement-based dependent cyto. Pag sinabi mo may nakitang complement, ang ibig sabihin po nito merong cell lysis. Okay? So complement-based cytotoxicity. So ibig sabihin this involves cell lysis. Okay? This procedure involves cell lysis. Okay? So, we have here anti-sera plus the patient lymphocyte. Kunyari, okay, so kunyari, we want to determine HLA-B27. Ito gusto natin malaman. We want to determine the HLA-B27. Okay, so yan ang gusto natin malaman. Yung HLA-B27. Remember, una, we need to determine if HLA-B27 is class 1 or class 2. HLA-B27 is class 1 kasi letter B. Okay, so HLA-B27, this is class 1 kasi this is letter B. Okay, so, since this is class 1, o syempre, saan nakikita ang class 1? So, ang class 1 pwedeng makita sa B cells and T cells. So tanong, how do we now separate the B cells from how do we now separate the lymphocytes from the other blood cells? So anong pwede natin gawin? So what we can do is to what? Perform the FICOL. Or remember yung FICOL. Okay? So yung FICOL, the density gradient centrifugation. Remember, we can use the density gradient centrifugation. Okay? So we can use the density gradient centrifugation. And the density gradient centrifugation can separate the B cells and the T cells from each other. Okay? So, ayan. So, we can use, what if I want to determine HLA-DR3? So, gusto natin malaman. HLA-DR3. So, kunyari, okay, we want to determine HLA-DR3. So, pagdating sa DR3, this is class 2. Okay? So, HLA-DR3, I repeat, this is class 2. Okay? So, this is class 2. So, since this is class 2, ang hanapin lang natin dito is what? Okay? So, take note, since this is class 2, si B cell lang dapat ang meron. Okay? So, I repeat, since this is class 2, si B cell lang ang meron nito. So, how do you now separate the B cell from the T cell? So, kung kanina, density gradient centrifugation can separate the lymphocyte from other blood cells. Dito, ang pwede natin gawin is the use of nylon wool. Okay? The most common way to separate the lymphocyte from other blood cells is through the use of a nylon wool. So the most common way to separate the lymphocyte from other blood cells, I repeat, is through the use of a nylon wool. Okay, so B cell has a tendency to attach to the nylon wool. Okay, so B cell, I repeat, it has a tendency to attach to the nylon wool. Okay? So it has a tendency, so B cell has a tendency to attach to the nylon wool. Okay, next. So ito na. So take note, ano yung specimen natin dito? If you want to detect class 1, then you can use both the B cell. If you want to detect class 2, then you use the MHC class to the B cell. Okay? So we have here anti-sera. So ano ba, kung gusto mo detect sa HLA-B, edi use anti-HLA-B27. If you want to detect HLA-DR3, then you use the anti-HLA-DR3. Okay? So the anti-sera plus the patient lymphocyte. So we have here the anti-sera plus the patient lymphocyte. Okay? So first, syempre after the addition, we now incubate. Okay? So after the addition, we now incubate. Okay? So after the addition, we now incubate. So dito sa incubation, dito po didikit yung antigen sa yung anti-sera sa antigen kung meron man. Okay? So diyan didikit yung anti-sera kung meron man. Okay? So binding of the antigen occurs. Okay? Complement is added. So kindly take note, the complement is added. So once na dumikit yung complement, ayan. Okay? So since there is antigen-antibody reaction, yung complement, didikit, pwede na nating i-add. So take note, pag may antigen-antibody reaction, take note, kung may antigen-antibody reaction, okay? So ano na mangyayari dito if there is antigen-antibody reaction? The complement will be activated. Okay? So the complement will be activated, but once it is activated, okay? Ano mangyayari dito? There will be lysis of the what? There will be lysis of the lysis of the cell. Okay? So, pag malyse yung cell, take note. Okay? Pag malyse yung cell, mabubutas si cell. Okay? So, halimbawa, nagkaroon ng lysis. So, mabubutas si cell. Okay? Pag mabutas si cell, you can now add, okay? O, pag mabutas si cell, you can now add the dye. Okay? So, once na mabutas si cell, I repeat, you can already add the dye. Okay? So, again, once na mabutas si cell, you can already add the dye. So, ano mangyayari? If you have the particular, okay, dead cells will able to get the dye. Okay, so dead cells will get the dye. Kasi syempre nabutas na sila eh. Syempre nabutas na sila, di ba? Okay, so dead cells will be able to get the dye. Okay, and take note, live cells cannot take up the dye and they remain colorless. So anong result dito? Okay, so anong mangyayari dito is, in short, may stain po si cell kapag positive. So take note guys may stain si cell kapag positive. Okay? So may stain si cell kapag positive. Pag nabutas po si cell, makakapasok po dyan si dye. So kapag siya po ay na-stain, ibig sabihin positive po yan. That is actually a positive result. Okay? So kapag na-stain, so positive result po yan. Okay? Next, we have here your genotyping. Okay, so we have here your genotyping. So, HLA genotyping, this is PCR-based amplification. Okay, so this is PCR-based amplification. And cells do not need to be viable in order to obtain the DNA. So, cells need not to be viable to obtain the DNA. Kanina kasi guys, ang complication, okay, so kanina, ang isa sa, how do we call it? Isa sa limitation ng phenotyping is that it requires live cell. Okay? So take note, ang isa sa limitation ng phenotyping kanina is that they require live cell. Okay? So buhay na cell. Bakit? Remember, we are actually testing the viability. Okay? So we are actually testing the viability of the cell. So kung buhay o namatay ba siya, kasi di ba pag patay, it will take up the dye. Kapag buhay, it will not take up the dye. Dito guys, not necessarily a viable cell. So hindi nyo kailangan that the cell should be viable. Okay? Next, another limitation of the phenotyping is that it requires the separation of the lymphocyte. Okay? So it requires the separation of the lymphocyte. Pero pagdating it requires the separation of the lymphocytes. So those are the two limitations of phenotyping. Again, it requires live cells, viable cells. Second, it requires the separation of the different of the blood cells, particularly the lymphocyte. Okay? So dito, ang madali lang dito guys, this is PCR-based amplification. So ibig sabihin, mas specific po ito because we will be talking about the genetic material. Okay? So why is it important? So why is it important for us to perform HLA typing? So HLA typing is important because this is important in tissue or organ transplantation. This is the most common use. Okay? Most common usage of HLA typing. So, tissue and organ transplantation, paternity testing. Okay, so, actually, this will just exclude, kung paternity testing, this will exclude the father. Kung may nagsasabi na, ay, ikaw yung tatay, you can ipapaternity test mo. Okay? Others, establishing risk of developing diseases and for racial ancestry and migration. Okay? Disease association, ito yung risk na sinasabi natin. Okay, so disease association, ito yung risk na sinasabi natin na if you have this particular HLA, then you have high risk of having this particular condition. For example, HLA-B27. Kung ikaw may HLA-B27, then you have high risk of having ankylosing spondylitis. Okay? So kung ikaw ay may HLA-B27, then you have high risk of having ankylosing spondylitis. Okay? So HLA-DR3, DR2, 4, and 3, examples lang naman, high risk of having multiple sclerosis, DR2, DR3 SLE, DR4 RA, and DR3, DR4 type 4 type 1. Again, risk lang po ang pinag-uusapan natin dito. I'm not telling you na pag meron ka nito, pag meron kang HLA na ito, you have this particular condition. No, we are only talking about the risk. Okay? Also, these are some of the organs that can be transplanted. The most common transplanted organ is the kidney. Okay, this is the most common. And take note, the safest to be transplanted, that is your cornea. So what is said to be the safest to be transplanted? That is your cornea. Sir, bakit cornea ang safest to be transplanted? Number one reason, it is avascular. Okay, so walang blood vessel, walang white blood cell. So take note, cornea is avascular, walang blood vessel, walang white blood cell. Okay, kumbaga walang security guard na magde-detect na that antigen is foreign. And second, it has low MHC class 1. Okay, low MHC class 1 and a total absence of MHC class 2. So, a total absence of MHC class 2. So, these are the reason why cornea is the safest to be transplanted. Next, we have your rejection reaction. Bakit may rejection? We have here your hyperacute rejection. So, pag maling organ ang binigay, ibig sabihin they are not incompatible, it will result to rejection. Ibig sabihin the immune system will reject the foreign antigen, the organ, kasi it contains foreign antigen. So, hyperacute rejection occurs minutes to hours. Hyperacute is usually associated with a preformed antibodies. Ibig sabihin ng preformed, may nabuo na before ibigay yung organ. And a very good example of this is the ABO. Okay? Always remember ABO is the most important blood group, and also we need to put into consideration when transplanting an organ to a patient. Because kailangan the patient should be ABO compatible. So the patient should be ABO compatible to the donor. So that is hyperacute. So preformed, remember, this occurs, nangyayari po ito kapag there is already the presence of a preformed antibodies. Okay? So we have here your accelerated, days, acute, days to months, chronic, 1 year or more, and graft versus host disease, we have 100 days or more. And we have here your mixed lymphocyte reaction. O pagdating sa mixed lymphocyte reaction, you will just be mixing the patient lymphocyte and the donor's lymphocyte. So pag nagalit si patient lymphocyte, pag siya po ay nagalit, okay, at dumami si patient lymphocyte, then it means the two lymphocytes are incompatible with each other. So hindi mo pwedeng ibigay yung organ. Okay, and this is, take note, this is actually, okay, a very good representation of allo recognition. So this is actually a very good example of allo recognition, the ability to recognize self from foreign antigen. Okay?
Okay, so now let's talk about the antibodies or the immunoglobulins. Kindly take note that aside from being called antibodies or immunoglobulins, these proteins are also referred to as your gamma globulins. So why are they called gamma globulins? Gamma globulins, because during serum electrophoresis, they are actually found in the gamma region. I repeat, they are referred to as your gamma globulins because during serum electrophoresis, they are actually found in the gamma region. But kindly take note, when referring to antibodies, the term gamma globulins is actually or is seldomly used. Why? Because not all antibodies are found in the gamma region. And a very good example of an antibody that is not found in the gamma region, that is your IgA. Hence, the term gamma globulins is only or seldomly used when referring to the protein antibodies. So when we are talking about antibodies, it is mostly commonly referred to as your immunoglobulins. Take note, the antibodies or immunoglobulins, generally speaking, these are the products of antigenic stimulation. So bakit ito nagawa? It's primarily because we were exposed to the foreign antigen. And as a result, we develop antibodies against this particular foreign entity. And kindly take note that this is the major humoral branch, a major component of the humoral branch of the immune response. Historically speaking, The basic structure of the antibodies was actually demonstrated and explained by Gerald Edelman and Rodney Porter. So I repeat, the basic immunoglobulin structure, it was explained by Gerald Edelman and Rodney Porter. So what are the functions, the primary functions of the antibodies? The antibodies, they primarily function in cell cytotoxicity. In other words, take note, cytotoxicity, in other words, cell killing. Okay, so cell cytotoxicity or cell killing. Take note, if, for example, this is your particular cell, and the antibodies will attach to this one. Okay, it will attach to this particular um, cell. So what will happen to the cell? Since there is antibody attached, complements can be activated. Remember, if the complement proteins are activated, it will cause cell lysis. What else? The bound antibody here can actually also trigger other cellular component. Example, it can trigger the NK cells or the natural killer cell to kill this particular cell. Okay, so those are just some of the mechanism wherein antibodies can trigger cell cytotoxicity. Also, antibodies, particularly those large antibodies like the IgM, they can cause agglutination. Also, antibodies can cause viral or toxin neutralization. And also, antibodies can be an opsonin. Remember, when we say opsonin, these are products, these are actually substances that causes or that promotes phagocytosis because opsonin will immobilize the organism. So opsonins can actually immobilize the organism, hence it can trigger the phagocyte to kill this particular cell. So these are just some of the functions of the antibodies. As what was mentioned earlier, the antibody structure were explained by Gerald Edelman and Rodney Porter. So Gerald Edelman was working at the Rockefeller Institute in the US, whereas Rodney Porter is actually working at the Oxford University in England. These two individuals are the one who explained the antibody structure. So let's talk about the basic structure of an antibody. So this is your typical structure of an antibody. As you can see, take note, there are two identical heavy chain. So take note, this red component here, this is your heavy chain. And as you can see, there are two identical heavy chain and there are two identical light chain. So this component here, this is actually your light chain. Take note, the middle portion here, this is your hinge region. And the hinge region, it actually gives flexibility to the antibody. Okay, so the hinge region, I repeat, it gives flexibility to the antibody. So take note, again, review, this is the heavy chain region. This is your variable, this is your light chain region. Next, let's talk about the domains. Okay, let's talk about the domains or regions of an antibody. So let's talk about the domains or regions of a particular antibody. So this black portion here, this is known as your variable region. It is called variable region because the amino acid sequence here is actually variable or changing. Okay, so we have here the variable region of the heavy chain, variable region of the heavy chain, and we have a variable region of the light chain, and another variable region of the light chain. Kindly take note that the variable region, this is said to be the first 110 amino acid. Okay, so this is said to be the first 110 amino acid. If this is the first amino acid up to the 110, that is said to be the variable region. Okay, so the first up to the 110, that is your variable region. In Tergion, it was mentioned that it is said to be the first 120 amino acid. Okay, so kay Tergion naman, it's the first 120 amino acid. So if ever this will be asked in the exam, so I will suggest the first 110 because it was also mentioned by Henrys that the first 110 is the variable region. Again, variable region because the amino acid sequence here is variable, and kindly take note, it is actually found in the amino group. It is found in the location of the amino group of the antibody. The opposite of the variable region is here the we have here the constant region. It is called constant region because the amino acid sequence here is actually repeating or constant. Okay, so the remaining amino acid aside from the variable region, that is your constant region. Okay, as you can see, the constant region here, take note, we have here the constant region of the light chain, we have the constant region of the heavy chain. So take note, the constant heavy chain region, it's actually long. Okay, it's still long, so it is furtherly divided into, take note, the constant heavy chain region 1, we have the constant heavy chain region 2 here, and we have here the constant heavy chain region 3. Okay, so 1, 2, 3. This is your typical immunoglobulin. But take note, there are some antibodies wherein there is an extra domain, and that extra domain, that is your constant heavy chain region 4. So I repeat, the extra domain, okay, so I'll put here an extra. So the extra domain is your constant heavy chain region 4. And what immunoglobulin contains this particular extra region, that is your IgM, okay, and IgE. So again, these two immunoglobulins are the immunoglobulins that has an extra domain. We have the IgM and we have here the IgA. The extra domain, I repeat, that is your constant heavy chain region 4. So let's discuss further the structure of the immunoglobulin. Kindly take note, the heavy chain, it determines the immunoglobulin class. I repeat, the heavy chain, it determines the immunoglobulin class. So when we say IgG, IgG means it has the gamma heavy chain. When we say IgM, IgM has the mu heavy chain. IgA, it means it has the alpha heavy chain. IgD means it has the delta heavy chain. And IgE means it has the epsilon heavy chain. So take note, the letters in an immunoglobulin corresponds to the heavy chain. And the heavy chain is very important because the heavy chain, it determines the immunoglobulin class. I repeat, for IgG, it has the gamma heavy chain. For IgM, the mu. IgA, the alpha. IgD, the delta. And IgD, you have the epsilon heavy chain. Next, we have the light chain. In the light chain, take note, there are two types of light chain. We have the kappa and we have the lambda. Okay, of the total percentage, it's 65% kappa, while the remaining 35%, it's the lambda light chain. Hence, the ratio of kappa to lambda is 2:1. Take note, the ratio of kappa to lambda, that is 2:1. Ang palatandaan ko dito, there are two p's, okay, 1 and 2 to 1b or 1d. So hence, the ratio of the kappa to lambda is 2:1. Next, what connects the heavy chain to heavy chain or heavy chain to light chain? That is your disulfide bond. Remember, this is your typical antibody. We have the heavy chain, okay? We have the light chain, light chain. Heavy chain to heavy chain linkage, that's normal. Heavy chain to light chain linkage, again, that is also normal. So this is heavy chain linkage. This is heavy chain to light chain linkage, okay? And these are normal. But if it will be light chain to light chain, so kumbaga, we will be combining these two. So light chain linkage, that is now an abnormal combination. Okay, so I repeat, that is now an abnormal combination. And a very good example of this light chain linkage, that is your Benz-Jones protein. Okay, a very good example, I repeat, of this particular protein wherein there is light chain linkage, that is your Benz-Jones. Remember, the Benz-Jones, it will coagulate or precipitate at temperatures between 40 to 60 degrees Celsius, whereas it will dissolve at 100 degrees Celsius. Okay, so I repeat, the Benz-Jones protein is an unusual protein seen in patient with multiple myeloma. I repeat, this is seen in cases of patient with multiple myeloma. Okay? And this protein will coagulate at 40 to 60 degrees Celsius. Ilagay mo sa 100, it will be dissolved. You return the temperature back to 40 to 60, then take note, it will again coagulate. So again, that is your Benz-Jones protein seen in cases of multiple myeloma or any other plasma cell disorder. And when we say hinge region, this is said to be the flexible region. This is the primary reason why the antibodies or immunoglobulins are considered flexible. So I repeat, the hinge region, this is the primary reason why the antibodies are considered flexible. It's primarily because of the hinge region. It is said to be rich in amino acid proline. Okay, I repeat, this particular protein is said to, region is actually rich in the amino acid proline or proline. So kindly take note of these different antibody variations. When we say isotype, it determines it is variation in the heavy chain. Okay? When we say allotype, this is actually variations in the constant region of both the heavy chain and the light chain. And lastly, we have here the idiotype. So when we say idiotype, this is actually the variation in the variable region of both the heavy chain and light chain. So I repeat, isotype, we are talking about the heavy chain. So if this is your isotype, take note, this is variations in the heavy chain. So itong part na to is the one associated to the isotype. Allotype, on the other hand, if this is your typical antibody, variations in the constant region. Take note, constant region of both the heavy chain and light chain. So ito naman yan. And lastly, idiotype, okay, if this is your antibody, there is variations in what? Variations in the variable region of both the heavy chain and the light chain. So kindly take note, this is actually the properties of an immunoglobulin. So I repeat, this is your typical properties of an immunoglobulin. So this table summarizes all. Kindly take note, the most abundant antibody, if we are talking about in the serum, okay? So the most concentrated, yung pinakamadami, tandaan nyo guys, GAMDE. Okay? So tandaan nyo GAMDE. The most abundant antibody in the serum, that is your IgG. Okay? Followed by IgA, followed by IgM, followed by IgD, and the least the abundant immunoglobulin, that is your IgE. Okay, so ulitin ko, the most abundant antibody in the serum, that is your IgG, followed by A, followed by by M, by D, and by E. Okay, so yung least abundant po dito, kindly take note, it's the IgE. Kung ang pinag-uusapan naman ay secretion, okay, kung secretions po ang pinag-uusapan, then the most abundant antibody there is your IgA. Okay? So the most abundant antibody is your IgA in the serum. So take note, GAMDE. So again, this summarizes the differences between the different immunoglobulin. By the way, this is coming from Stevens. So let us discuss further. So first we have the IgG. IgG means it has the gamma heavy chain. So take note, IgG, as what I mentioned, this is the most abundant antibody in the serum. Hence, this is said to be the major antibody in the blood. Okay? What else? This is produced during the secondary immune response. So I repeat, this immunoglobulin here is actually produced during the secondary, okay? This is produced during the secondary immune response. Okay, so again, this is produced during the secondary immune response. So later on, we'll be talking about the immune response, meaning this is the immune system reaction to a particular antigen. This is known as an antibody that can cause complement activation via the classical pathway. Pero kindly take note that for the IgG to activate the complement, it requires two molecules of IgG. I repeat, for this particular um, antibody to activate the complement protein, it requires two molecules, okay? Two molecules of IgG to activate the complement. And take note, since this is the smallest, kindly take note, this is the smallest, considered as the smallest antibodies. Hence, this is the only antibody that can pass through the placenta. Hence, IgG is one of the antibodies that can be passively transferred to the fetus. Okay, so passively transferred to the fetus because the IgG can actually what? Pass through the placenta. What else? In all of the immunoglobulin classes, IgG is the one with the longest half-life. I repeat, in all of the different immunoglobulin classes, IgG is the one with the longest half-life. Particularly, the half-life for this particular immunoglobulin is actually 23 days. So what are the major functions of the, okay? So what are the major functions of the IgG? Okay, so number one, it provides immunity. It provides immunity for the newborn because IgG is the only antibody that can cross the placenta. What else? As what I mentioned earlier, the IgG can actually activate the complement. If the complement is activated, remember, it will result to cell lysis. What else? It can coat, since this is the smallest immunoglobulin, I repeat, this is considered as the smallest immunoglobulin, it actually coats the organism. Okay? But when the organism is actually coated, then it can act as a opsonin. Okay? So, it can act as an opsonin. Okay? What else? It can neutralize toxin and viruses. And this also participates in the agglutination and precipitation reactions. I repeat, this participates in the agglutination and precipitation reactions. So again, these are the major functions of the IgG. Also, kindly take note that there are subclasses. As you can see, all the subclasses, okay, all the subclasses, the they have the same heavy chain. Okay, all the subclasses, I repeat, they have the heavy chain. Okay, they have the same heavy chain. Okay, so they have the same heavy chain, so gamma. So as you can see, so same heavy chain. So what will now be the basis of differentiation? So differentiation is actually based on the disulfide bonds. I repeat, differentiation of these particular subclasses are actually based on the disulfide bonds. Okay. Again, immunoglobulin subclasses are differentiated based on the location and presence, the location and numbers of the disulfide bonds. Kasi all of them, they have the same heavy chain. Okay? So I repeat, all of them, they have the same heavy chain and they are differentiated based on the location and numbers of the disulfide bonds. Take note, what is best here, okay? IgG is the best, IgG1 is the best subclass that can, this is the best IgG subclasses that can pass through the placenta. Whereas IgG2, okay, so IgG2, this is the immunoglobulin that can actually what? That is the one that has difficulty in passing through the placenta. So the best, actually there is a pattern here. This is the best. Ito po yung kulelat. This is the best in complement activation. Whereas IgG4, ito naman po yung kulelat. Okay, wherein it has a difficulty in passing through it has a difficulty in activating the complement. Okay, so the best in passing through the placenta, the kulelat, the best in activating the complement, kulelat. Okay, so this is your typical IgG subclasses. Okay, so again, subclasses are differentiated based on the location and the number of the disulfide bonds. Next is we have the IgM. If the IgG is the smallest antibody and it only coats the organism usually, we have the IgM. The IgM naman, this is considered as the largest immunoglobulin, and this is an antibody that can actually cause agglutination. I repeat, this is an immunoglobulin that actually causes agglutination. So take note, this is the major class of antibody secreted into the blood in the early stages of the primary antibody response. Remember, the IgG is the one secreted in the secondary immune response. What else? This IgM is commonly, the serum IgM is a pentameric IgM. So when we say pentameric IgM, this is an antibody that is made up of 10 antigen, made up of 5 monomers. They are actually linked by a joining chain. So if this is your typical serum IgM. As what I mentioned, this IgM is actually made up of 5 monomers linked by a joining chain. So this ring here represents a joining chain. And take note, there are actually 10 binding site. I'm referring to the typical serum IgM. And this is said to be the first class of antibody to be produced by the developing B cells. Remember in the previous discussion, the IgM is the first immunoglobulin that is actually produced in the development of the B cell. So since IgM, I repeat, is the largest antibody, this IgM can actually cause agglutination. This is also referred to as your macroglobulin. Why macroglobulin? Because this is an antibody that is actually big. It can cause agglutination because of its size. So what are the functions of the IgM? So IgM, this is said to be the best complement fixer. Why is IgM the best complement fixer? Because remember, it only requires one IgM or one molecule of IgM to activate the complement. I repeat, it only requires one molecule of IgM to activate the complement. What else? Since this is the largest antibody, it can cause agglutination. Okay, it can also, just like IgG, it can cause opsonization. And take note, it can also cause neutralization of the organisms or any toxins or other substances. Kindly take note that the pentameric form of the IgM is found in the serum, whereas the monomeric form is found in the surface of the B cell. So I repeat, the pentameric form is found in the serum, whereas the monomeric form is actually found in the surface of the B cell. Another immunoglobulin is the IgA. If the IgG is the major antibody in secretion, IgG is the major antibody in the serum, IgA, on the other hand, is the major antibody in the secretion. Since this is the major antibody in the secretion, this is said to be the antibody that is in charge in mucosal immunity. I repeat, since this is an immunoglobulin that is found in the secretion, I repeat, this is the immunoglobulin that is responsible for mucosal immunity. This is an immunoglobulin associated with anaphylaxis. And kindly take note, this is an immunoglobulin that has an anti-inflammatory agent. Okay, so when we say secretions, this could be present in the milk, saliva, tears, and also respiratory or intestinal secretion. Kindly take note, just like IgG, IgA can also be passively transferred to the baby. Okay, in the case of the IgG, it can be passively acquired by the fetus via the placental transfer. Okay, IgA, on the other hand, is passively transferred to the baby via the breast milk. Hence, the breast milk is best for babies. So there are two forms of IgA. The IgA type 1 is a monomer. Okay, so IgA type 1 is a monomer. Okay, this is the major antibody in the serum. Whereas the IgA type 2, this is your dimeric antibody. And this is the immunoglobulin, the IgA, that is found in the secretion. So type 1 is a monomer. Type 2, on the other hand, it is a dimer. Okay, so just like the IgM, in a dimeric IgA, it is actually joined by a J chain or joining chain. And aside from the presence of the joining chain, it has the secretory component, or sometimes referred to as your secretory piece. I repeat, secretory component or secretory piece, they are actually the same. So what is the function? So what are the functions of the secretory component or piece? Okay, so the secretory component or secretory piece, it actually, number one, facilitates in the transfer of the IgA to the secretions. I repeat, number one, it facilitates the transfer of the secretory IgA or the secretory IgA or the type 2 IgA to the secretion and to the secretion. So facilitates transfer. And number two, it prevents enzymatic degradation of these particular antibodies. I repeat, it prevents enzymatic degradation of this particular immunoglobulin. Next, another immunoglobulin is we have the IgD. The IgD, the major function is immunoregulation. Take note, in all of the immunoglobulin, IgD is said to be the most sensitive to enzymatic degradation. I repeat, in most IgD, this is said to be, in most immunoglobulin rather, this is said to be the most sensitive to enzymatic degradation. This is the major membrane found on the surface of a high proportion of B cells. Particularly, this IgD is actually found in a mature B cell. Remember in the previous discussion, if it's only IgM, that is an immature B cell. But if it's an IgM plus IgD, then that is now present in the mature B cell. And this immunoglobulin is postulated to be, or an antibody that has an anti-idiotypic antibody. The IgE, take note, this is among the immunoglobulin, this is the least in terms of concentration in the serum. And aside from being the least immunoglobulin in the serum, this is said to be the most heat labile antibody. Okay, so this is said to be the most heat labile antibody. And this antibody has the ability to attach to human skin. Hence, this is said to be a homocytotropic antibody. And this can actually initiate allergic reaction. Hence, this is said to be a reagenic antibody. So take note, this antibody has the ability to attach to the human skin. And this can initiate allergic reaction. Additionally, IgE has high affinity to basophil and mast cells. So I repeat, additionally, IgE has high affinity to basophil and mast cells. Kaya later on, once we discuss the hypersensitivity reaction, you will know that the IgE is the what? The IgE is the primary mediator of the type 1 hypersensitivity reaction. And syempre, what cell will help the IgE? That is your basophil and mast cells. And take note, this IgE said provides a striking example of the o ibig sabihin ng bifunctional, ibig sabihin ng bi, di ba ibig sabihin yan 2. So bifunctional nature of the antibody. So take note, the Fc portion binds to the target cell, whereas the Fab portion, it binds to the allergen. So Fc means fragment crystallizable, Fab means fragment antigen binding. So Fc, okay, is the one that binds to the target cell, the fragment crystallizable, whereas the fragment antigen binding, it is the one that binds to the allergen. So this is your typical immunoglobulins. Okay, so IgG and IgD. Okay, this dimeric IgA, this is your secretory IgA. So I repeat, this dimeric IgA is your secretory IgA. And we have here the pentameric IgM. It is made up of 5 monomers. Okay, oh, by the way, the basic structure of an immunoglobulin is a monomer. Okay, ito po yung mga monomers. Ibig sabihin ng monomers, isa lang. Okay, monomer. This one is a dimer. Okay, and we can say this one, aside from being called pentamer, this can be mentioned as your polymer. So, ibig sabihin, it is made up of more than 2 monomer because it's made up of 5 monomers. So, I repeat, this is a polymer. Take note, the best indication of a current infection is a fourfold rise in the antibody titer. So when comparing 2 serum samples collected from a patient during the beginning and the later stages of the infection. So how will you know that there is a current infection? There is actually a fourfold rise in the antibody titer. So I repeat, there is a fourfold rise in the antibody titer. Next, we have here the fragmentation of a monomer. So monomers can be fragmented. So one of the enzyme that can cause fragmentation is number 1, we have the papain. So the papain cut monomers exactly at the hinge region, and monomer is cut into 3 parts. So what are those 3 parts? We have here the Fab1, the Fab2, and we have the Fc portion, the fragment crystallizable. So when we say Fab, it's made up of 1 light chain and 1/2 of the heavy chain. So I repeat, so Fab means it is made up of 1 light chain and 1/2 of the heavy chain, and they are held together by the disulfide bond. When we say Fc, that is your fragment crystallizable, it represents the carboxy terminal halves of the 2 heavy chains. So Fc fragment, it represents the 2 carboxy terminal halves of the 2 heavy chain. So sabi nila, so pag papain, the monomer is cut into 3 parts. So tandaan nyo lang daw, syllables. Papain. Okay, so papain, 3 syllables, 3 parts. And another reagent that can fragment a monomer, we have the pepsin. And pepsin, it cuts the monomer below the hinge region, and monomer is cut into 2 parts. What are those 2 parts? We have here the Fab here, and we have the Fc. Okay, so fragment, crystallizable, and the Fab. Next, we have here the affinity. So affinity is defined as the initial force of attraction between a single Fab and a single epitope. So I repeat, affinity is defined as a single. This is defined as a initial force of attraction between single Fab and a single epitope. Okay, so when we say single Fab, when you say Fab, remember this is your fragment antigen binding. When you say epitope, this is your exact antigen binding site. So affinity is the initial force of attraction between a single Fab and a single epitope. Okay, so again, this is the initial force of attraction between a single Fab and a single. Remember when you say epitope, this is your exact antigen binding site. So epitope, this is your exact antigen binding site. And what connects this single Fab with a single epitope? What are those? What are those? What are those forces that actually attracts or connects the antigen and the antibody? We have here the ionic bonds, uh, hydrogen bonds, hydrophobic bonds, van der Waals forces. Basta a single interaction, it is referred to as your affinity. Next, avidity, on the other hand, this is now the sum of all attractive forces. So avidity is the sum of all attractive forces between an antigen and an antibody. So avidity, I repeat, is the sum of all attractive forces between an antigen and an antibody. Remember, um, avidity can be remembered as your multiple affinities. So I repeat, avidity can be remembered as multiple affinities kasi pag isa lang yan, if there's only one interaction, that is affinity. But if there is multiple affinities, then it is now referred to as your avidity. And remember, if there is increased avidity, malakas ang kapit sa isa't isa. Okay, and there it means what? Kung malakas ang kapit sa isa't isa between the antigen and antibody, there is a decreased tendency for the complex to dissociate. Okay, so there is a decreased tendency for the complex to dissociate. Okay, so decreased tendency for the complex to dissociate. Okay, next we have here your immune response. Okay, so immune response. The immune response, this is your immune system reaction to a foreign entity. So immune response, I repeat, is defined as your immune system reaction to a foreign entity. So there are four parts. Okay, so there are four parts. We have here the log, we have here the log, we have your stationary, and we have here your decline phase. So pagdating sa lag phase, okay, ito po yan. Pagdating sa lag phase, okay, in the lag phase, there's no antibody production yet. Okay? In the lag phase, take note, there's no antibody production yet pagdating sa lag phase. Okay? Pagdating sa log phase, okay? So pagdating sa log phase, ayan. Log phase, there is already antibody production. Okay? So pagdating sa log phase, I repeat, there is already antibody production. So lag, wala pa. Log, there is already antibody production. And we have here the stationary phase. And the stationary phase also referred to as your plateau phase. In the stationary or plateau phase, ano naman meron dito? The antigen concentration, the antigen production is equivalent to your antibody. I mean, the antibody production rather is equivalent to antibody degradation. So quits lang, may nabubuo, merong nasisira. And pagdating sa decline phase, take note, pababa na po si antibody. Okay? Pababa na kasi, mas madami na po. There are more antibodies that are destroyed as compared to antibodies that are actually produced. So that is now your decline phase. Okay? So ayan. So dito guys, in the immune response, the highest titer of the antibody is actually found in the stationary phase. So I repeat, the highest titer. So the highest titer of an antibody is actually found in the stationary phase. So the highest titer, I repeat, is found in the stationary phase because that is the highest peak. So stationary phase, I repeat, that is the highest peak. Hence, you will be seeing the highest titer in the topmost, and that is your stationary or plateau phase. Okay, so there are two types of immune response. We have here the primary immune response and we have your secondary. So the moment that you are exposed to the antigen, so the moment that you are exposed first to the antigen, that is your primary immune response. It will result to the primary immune response. So anong meron doon? So sa primary immune response, okay, first time na I'm exposed, it is associated with a longer lag. Whereas secondary, the second time that you are exposed results to a shorter lag. So longer lag phase primary immune response, shorter lag phase pagdating sa secondary immune response. Okay? So pagdating sa secondary immune response. Okay? So bakit ganun, sir? Because of memory. Okay? Bakit ganun? That's primarily because of memory. Remember, the cells of the acquired immunity, they have memory. They have the ability to remember a prior exposure. So they have, I repeat, the ability to remember a prior exposure. Okay? So kaya memory. So they have memory. Kaya shorter lang naman po. Next, in terms of antibody production, mas madami po yung mapoproduce na antibody sa secondary immune response, okay, as compared to the primary. Okay, so mas madaming antibody na bubuo sa primary immune response, I mean sa secondary as compared to the primary. Actually, you can think of this as what? As the first and the second dose of vaccine. Okay? Yung primary immune response, that's your first dose. Secondary immune response, that is your second dose. Bakit? Sabi nila, mas protektado ka, di ba, nung kasagsagan ng pandemic. You are protected if you are actually protected if you are already given the second dose as compared to the first dose. So, pag na second dose na, why? Because in the second dose, in the secondary immune response, more antibodies are actually produced as compared to the antibodies produced in the primary immune response. Okay? So yung primary, that is your first, that is actually your first dose. Okay? So longer, shorter lag, less antibody production, more on antibody production. What else? There is an abrupt decrease, mabilis yung pagbaba niya. Ito, there is a slow decline in the antibody concentration. So there is, I repeat, a slow decline in the antibody concentration. Okay? So ayan, and the major, aside from this one, is we have here your immunoglobulin classes. Okay, so we have here your different immunoglobulin classes. For primary, it's actually IgM. For secondary, it's actually IgG. Okay, so primary, it's IgM. Secondary, it's IgG. Okay, to summarize, these are your differences between what? To summarize, these are your differences between your primary and secondary immune response. So primary, this is the first time that you are exposed to the antigen. So primary, I repeat, this is the first time that you are exposed to the antigen. Pagdating sa secondary, this is your second exposure. Okay, so second exposure to the particular antigen, or also referred to as your anamnestic response. So secondary, I repeat, is referred to as your anamnestic immune response. So this is an illustration of our primary and secondary immune response. Dito guys, take note, I want to emphasize the function of, okay, so I want to emphasize the function of the memory cell. Remember, ang trabaho po nito memory cell, this is important in the, this is a backup or a reserve cell for the secondary immune response. So I repeat, this acts as a backup or reserve for the secondary immune response. Yung sinabi ko last time, not all antibody, not all cell will be activated. Okay, not all cell will be activated. Okay, not all cell will be converted to become the plasma cell. Some will become memory cell. And this memory cell will be useful in the secondary immune response. Okay, repeat. This cell, I repeat, the memory cell will be useful in the secondary immune response. Okay, so secondary immune response, it will give you larger clone cells. Okay, larger clone cells means more antibody production. So more antibody production. Also, we have here the monoclonal antibodies. So meron tayong tinatawag na monoclonal antibodies. These are purified antibodies cloned from a single cell. This is usually used in the analysis of cell membrane antigen. So the monoclonal antibodies, this was discovered by Kohler, okay, Milstein, and Journey. Okay, so this was discovered by Kohler, Milstein, and Journey. So usually this is actually used in the analysis of cell membrane antigens. So what is the procedure that is used in the production? So what is that specific procedure that is actually used in the production of what? A procedure that is actually used in the production of monoclonal antibody. We have here your hybridoma technology. Okay, and the hybridoma, why is it called a hybrid? From the name itself, hybrid, because this is a combination of myeloma cells and the spleen cells. So this is called a hybrid, I repeat, because this is a combination of what? Okay, a myeloma cells and the spleen cells. Okay, so myeloma cells, spleen cells, ano po yung ginagamit natin dito? Mice. So what we use here is actually a mice. Okay, so mice is actually used in the production of this particular antibody. Okay, so for example, we want to prepare an antibody against the HLA-B27 antigen. So what we can use is what? So in the production of the HLA-B27 antibody, so we can use the anti-HLA-B. I mean, we can inject the HLA-B27 to the rats. Okay, so we can inject HLA-B27 to the rats, to the mice, and the HLA-B27 is foreign to the mice. Okay, so ayan. And the mice, kukunin natin yung spleen. Remember, the main site of antibody production, that is your spleen. So the spleen cells with the plasma cells will be fused with the myeloma cells for you to produce a hybrid. And yung hybrid na yun, guys, is the one responsible in producing the particular monoclonal antibody. Okay? So monoclonal, aside from for diagnostic purposes, monoclonal antibodies can actually be used as a treatment of cancer and autoimmune diseases. So monoclonal antibodies, I repeat, can be used as a treatment of cancer and autoimmune diseases. Yun nga lang, in terms of this, monoclonal antibodies, the main concern for this particular um, treatment is actually what? It is said to be highly immunogenic. Remember, this is made from animal sources. Okay, in most cases, this is actually used in animal sources. So hence, remember, the more foreign, the more immunogenic. So the more foreign, the more immunogenic. Okay. So what are those different monoclonal antibodies? We have the mouse. Okay, mouse ends with OMAB. The chimeric monoclonal antibody CMOB. Humanized, it ends with ZUMOB. And human, it ends with UMOB. Okay, so these are the different monoclonal antibodies. So my mouse, chimeric, humanized, and the human. Okay, so let's differentiate the different types of monoclonal antibodies. So first, we have here the mouse. Okay, first we have here the mouse. Tinawag siyang mouse because the whole component of the antibody is actually mouse-derived. Okay, and syempre, this is said to be the most immunogenic. Okay, this is said to be the most immunogenic kasi, as you can see, the whole components are actually made up of mouse. Okay, so the whole component, I repeat, is actually made up of mouse. Okay, chimeric on the other hand, so pag sinabi po natin chimeric, take note, this region here, anong region to ng antibody? That is your variable region. Okay, so yung chimeric, because this is actually a combination. the variable region is mouse-derived, whereas the constant region, it is actually human. Okay? So mouse, variable region, yung katawan, constant region, it is actually human-derived. Okay? Humanized, on the other hand, not all, okay, not all variable regions are made up are mouse-derived. So pag sinabing humanized, only a portion of the variable regions are actually, only a portion of the variable regions are actually made from or are actually mouse-derived. And when we say human, full po yan. Okay? When we say human, okay, it's full. It's made from, okay, it's when you say human, it is what? Human derived. So take note in terms of immunogenicity, ang immunogenicity po dito is what? The most immunogenic si mouse, the least immunogenic si human. Hence, these mouse-derived monoclonal antibodies are actually what? These are mostly used in as a form of for diagnostic purposes. Hindi siya as a form of treatment, but rather this is a form for diagnostic purposes only. Okay? So hindi po treatment for diagnostic purposes only as a form of diagnosing. Okay? So again, the most immunogenic si mouse, human, the least among this one. Another topic is we have here the cytokines. Okay, so we have here the cytokines. Cytokines. Cytokines are chemical messengers that regulates. These are chemical messengers that regulates the immune system. Okay, and the generic term for this one is that these are soluble mediators. Okay, so these are soluble. Generic term. Take note. These are actually soluble mediators of the immune system. Overproduction of the cytokine is referred to as your cytokine storm. So I repeat, the overproduction, pag nasobrahan dumami ng pag-produce ng cytokine, it is referred to as your cytokine storm. So overproduction of cytokine is referred to as your cytokine storm. Okay, so overabundance. Take note in our body, pag sobra-sobra, it's actually what? Too much production is not beneficial to our body. Okay, so too much production of cytokine is not beneficial. Actually, meron po yung bad effect to us. Okay. So in this particular session, we'll only be talking those cytokines that are actually significant or important. So we'll be talking about those cytokines that are actually significant or actually important. First, we have here the interleukins. Interleukins came from the word interleukocytes because this is a cytokine that functions primarily in immune cell differentiation and activation. So take note, I repeat, this interleukin came from the interleukocyte, primarily functions in immune cell differentiation and activation. So there are many interleukins, and we'll be focusing more on the important cytokines. Okay, so we have here your interleukin 1. Diba, in our previous discussion, interleukin 1, this is an endogenous pyrogen. Okay, so this is actually the one responsible for the fever. Okay, so endogenous pyrogen because this is the one in charge with the fever. Okay, what else? Interleukin 2, formerly known as your T cell growth factor. So formerly known as your T cell growth factor. And bakit former name po yung T cell growth factor nito? Because this is a cytokine that can activate both the B cells and the T cells. Kaya hindi lang po siya T cell growth factor because this can activate both the B cells and the T cells. And interleukin 6, it stimulates the B cells. So interleukin 6 can stimulate the B cells. Interleukin 2 is a former this is a former T cell growth factor. Okay, so former name is a T cell growth factor. So this important in the growth and differentiation of the B cells and the T cells. It induces the lytic activity of the natural killer cells. So it induces the lytic activity of the natural killer cells. Okay, interleukin 4, it regulates the T helper type 2 immune activity. So it help drives the antibody response. So interleukin 4, it help drives the antibody response. So this is important in the antibody. Remember the T helper type 2 is important in regulating antibody production. Hence, interleukin 4 is also important in the production of antibodies. Interleukin 6 stimulates the B cells to become plasma cell and induces the CD4 positive cells to produce cytokines. So again, interleukin 6 stimulates the B cells to become plasma cell and it induces CD4 positive cells to produce cytokine. And another important cytokine is we have here the interleukin 10. So interleukin 10 has an inhibitory effect. So interleukin 10, I repeat, it has an inhibitory effect to the immune system. Okay, so another example is we have here your interferons. Okay, so interferon came from the word interfere. So interferons, I repeat, it came from the word interfere And ano po yung purpose nito? They interfere in viral replication. Yung purpose po nito is that this interferes in viral replication. Okay, so this interferes, I repeat, in viral replication. They exert virus non-specific but host-specific antiviral activity. Yan, so that is your interferons.